So I'll start. Hello, everybody. My name is Shivani Sehrawat. I'm a master's student at University of Saskatchewan, Canada, and today I'm presenting my research work. So a little bit of background. Genotype is the component of cell's genetic composition, that is a DNA sequence that defines an observable trait, that is the phenotype. The genomic selection, it has emerged as an important tool which can utilize genomic information for modeling the crop yield for effective and rapid selection to meet the production challenges. Various genomic prediction models and algorithms, such as artificial neural networks, Bayesian estimation, and the most used conventional statistical predictive models are RR-BLUP and G-BLUP. However, these models they make strong assumptions that the random effect of genotype follow a normal distribution and do not consider the non-linearity between the variables. That is why they face challenges due to the high dimensionality of the genome-wide marker data and interactions between alleles. So, to overcome these challenges, deep learning models can be used to deal with large genomic data for genomic selection. The popular feature of deep learning is its high prediction accuracy using multi convolutional layers in the architecture of a neural network. So, SNPs, single nucleotide polymorphisms, are the most common type of genetic variations among people. Each SNP represents a difference in a single DNA building block called nucleotide. According to a number of recent genome wide association studies, only a few common SNPs are involved in most diseases, and these associated SNPs can only explain a small percentage of disease susceptibility. Structure variations represent an important part of genetic diversity in plants and play a role in phenotypic variation. They are complex genetic rearrangements that can result in gene duplication, creation of new genes, and nowadays, variation is also being acknowledged as a substantial source of genetic diversity, even though SNPs were once considered to account for the most selective variation in the genome. Structure variations, as opposed to SNPs, can produce large scale perturbations of cis regulatory regions, making them more likely to impact gene expression and phenotypes quantitatively. Transposable elements are also a source of genomic markers for modern plant breeding due to their widespread distribution and abundant genetic variation. A recent study has concluded through GWAS that TE markers have similar ability to discover association patterns to SNP markers. Earlier, plant genomic investigations were hampered by technological limitations and lack of high quality reference genome assemblies, which prohibited a detailed analysis of both structure variations and transposable elements in plants. But recent improvements in genomic technology, notably whole genome sequencing and long read mapping, offer the generation of high quality plant genome and pangenome assemblies, as well as to exposure to a diverse set of structure variations and transposable elements. So, the existing methods in genomic selection can only consider the SNP variants as genomic markers to predict the phenotypes. For these standard linear genomic prediction models, the number of genome wide SNP features largely exceeds the sample size, which leads to overfitting. But for deep learning models, studies have shown that these methods base their predictions on overall genetic relatedness rather than just on the effects of particular markers. So, this research demonstrates the need of looking further than SNPs when understanding evolutionary processes and how structure variations and transposable elements can help us comprehend uh, variation at the intraspecific level or during early divergence. So, the flowering plant Arabidopsis thaliana is an ideal plant species for researching genotype, phenotype, environment interactions because their naturally inbred strains allow for a repeated phenotyping and have adapted genotypes under a variety of control circumstances. So, this structure variant data set includes several types of structure variations such as deletions, duplications, and inversions. And the phenotypes of the match samples were downloaded from the Arapheno database. Orzia sativa rice, which is also called known as rice, is a monocotyledonous flowering plant that is one of the world's most significant agricultural plants, providing the primary source of nutrition for half of the world's population. The T and SNP markers were used from the given links, and the phenotypes used in the study are measured for the same 176 accessions from the study given in reference 3. Sorry. So, a NOV-GM Deep, it is a deep learning model for genomic selection using novel genomic markers. It is a one-dimensional convolutional neural network to predict phenotypes which are presented in the study. This model is trained separately on three different types of data: structure variation, transposable elements, and SNPs. Uh, the evaluation metric used here are PCC, mean absolute error, and then the standard deviation of mean absolute error. The model architecture, the figure here shows that the first layer has 16 filters of size 5 for all the next convolutional layers. The filter size is decreased by 2, and for the last two convolutional layers, uh, in order to reduce the dimensionality of the prominent features for of all the last convolutional layer, a 1D max pooling layer is used to after the CNN layers with a window size of 2 and a stride of 2. And then the data is transformed using a flattened layer and fed into the fully connected layer. Dropout with zero point 0.6 drop ratio is used before dense layer to drop some feature map from the convolutional layers to decrease the complexity of the model and to avoid overfitting. And then there is a once dense layer with one output neuron. So, the Arabidopsis thaliana is a diploid species and has genotype as 0 by 0, which means homozygous for reference allele, and 1 by 1, which means homozygous for alternate allele. The strategy shown here is used to represent genotype of inversions, duplication, and deletions. And the genotypes were transformed using one hot encoding binary arrays. In one hot encoding, given a data set with different features, the encoder find the unique values per feature and transform the data into a binary one hot numeric array. Then the data was fed to as a 3D matrix to the model. And or this similar strategy is used to encode transposable element and SNP variants shown here. This table here shows the data representation of different markers used in the study. Now let's talk about the results. So the model was trained with both structure variants and SNP datasets for Arabidopsis. So first we are discussing about the Arabidopsis results. From all the samples, 80% were used for training, 20% were used for testing using threefold cross-validation. Uh, Pearson correlation coefficient of metric is used for evaluation, which is a measure of the linear correlation between the observed and predicted phenotypes, whose value ranges from -1 to 1, indicating 1 indicating total positive correlation, a 0 indicating no correlation, and -1 indicating negative correlation. So you can see that the two statistical models and the deep learning models were evaluated with the same data to compare the overall prediction performance. And as shown in the table, phenotypes predicted by NOV GMD shows the strongest correlation and outperforms the other models. Moreover, phenotypes predicted by the model using structure variant data shows better results than ones predicted using SNP data. So this shows the effectiveness of structure variation data over SNPs. For the second data set, the proposed model was trained with both transposable element markers and the SNP data. Similar data split strategy was used. And though here the proposed model doesn't show better correlation than classical genomic selection models and also the deep learning models, but the correlation value predicted using transposable element is still better than the ones predicted using SNPs. The reason behind the low correlation values could be the small sample size, which was mere 176 samples, as the deep learning models work better when trained on large number of samples. So here, the predicted values and true values of phenotypes calculated from the proposed model were plotted on the testing sets of structure variant and SNP data for the Arabidopsis. And the orange line in these figures represent the trend of the relationship between the observed and the predicted phenotypes in blue points. Points close to the fitting line shows positive correlation, and far from the fitting line shows negative correlation. The graph here for in the figure A shows that the predicted values by the model positively follow the ground truth values with high confidence level, and the graph B shows positive correlation but with low confidence level, as there is more fluctuation for the SNP dataset. So mean absolute error metric is also used to calculate the validation or loss on the test set. The standard deviation of all the MAE values on the test set was also calculated to see the deviation among those values. So this can provide some insight into the model. A model with a low MAE indicates that it have a good average performance over the data set, and if the model also have a low standard deviation, then it tells that the performance is not only good on average but also uniform on the data set. So the low values of MAE and here show for the structure variation and the transposable element marker show the higher prediction accuracy of the NOV-GM dip model over the SNPs. The MAE was loss was watched to select the best model during the training process of 150 epochs with early stopping. The model with the lowest MAE was selected to evaluate the model. So in figure A, you can see the model is neither overfitting nor underfitting, as the training and in the training curve is lower than the validation curve, which shows the model is overfitting in the figure B. Similarly, here the model for the transposable elements in figure A is neither overfitting or not underfitting, but for SNPs, the model is overfitting. So the large number of features, that is the SNPs, expands the hypothesis space, making the data more sparse, and this could lead to the overfitting problems. So some conclusions. So the both the statistical models have low PCC values compared to the proposed model for the Arabidopsis data. Low PCC values for Orsia data for transposable elements, even negative for SNPs uh, because of the low sample size to train a deep learning model that is not recommended. It is important to apply deep learning model to large training and testing data sets. And due to a large number of SNP markers across the genome uses features, overfitting increased in the model, and this causing the small and large p problem. And the performance comparison with the deep learning models was only possible for SNP data, where the proposed model outperformed the other two. The study paves the way for the novel use of structure variations and transposable element markers in the field of genomic selection instead of just using SNPs. Here are the references. Thank you so much. So yeah, you guys can. I'm ready for questions. Sorry
Thank you.
about if it was a little long.
No, it's okay. Thank you, Dr. Giovanni. Is there any questions? Yeah, bueno, I have one. Do you think the specific whole genome duplication of plants have an impact in your model? I mean, Brassica have several rounds of whole genome duplications that, I mean, Carica papaya doesn't have these rounds of whole genome duplication. How this could impact the models to find these novel structural variations?
Uh, yes. So you're talking about that how whole genome duplication is can affect the novel genomic markers, if I understand it correctly.
Yes.
Yeah. So I haven't been to that area to so that's why the small genomes were taken, because it's a novel work right now, and I also couldn't find I've tried to look for different kinds of transposable element marker data sets, but it's really not available. And so I think a research is still going on in that area, and but that is a great idea to include whole genome duplication also, and maybe that could be a future work. Yeah.
Yep.
So I hope I answer your question. I mean, I really didn't answer, but like this could be a future work.
That's okay. Thank you.